Hello everybody, welcome to RahbarTalkShow. We want to discuss this session about the biological effects is so-called protein corona. This phenomenon can affect the bioactivity of any nano-sized particles inside our body, especially extracellular vesicle or exosome. So please follow us and see what happens. Hello, Farsin. Can you just explain what is a protein corona?
Yeah, actually, in the start of the session, I would like to say that the protein corona is a complex of proteins or some molecules that gather around any nanoparticle that enters the fluids of body. If we want to talk it in general, it's kind of, how to say, it shapes the nanomaterial which is in the blood or any fluid in our body to absorb it, to have any immunological effects on it. So it's kind of a cover that shapes the nanoparticle which enters the body.
Great, thank you. Mrs. Khosravi, how about you about protein corona?
As we talked last session about exosomes, when enter in a biological fluids like blood and serum, different kinds of proteins absorb onto the exosome surface and make an outer layer which is called PC or protein corona.
It's an abbreviated form of protein corona.
Yes.
Yeah. Great.
And this layer makes protein corona around the exosome.
You mean that it includes any nano-sized particles, as I understand? Great. Mrs. Shafrizan, and what's your idea about protein corona or PC?
Actually, when a nanoparticle enter in biological environment like blood, plasma, or even cell culture medium, the proteins, different kinds of proteins in this environment can absorb onto the surface of these nanoparticles. And if I were to summarize in one sentence, I can say that it determined the fate of nanoparticles in the body.
Great, thank you, thank you. Mr. Nazif Kerdar, do you think that only the component of protein corona are proteins, or?
Well, to the best of my knowledge, I think that there are lots of things can happen and absorb to the protein, to the nanoparticles drug while they enter to the body. One of them is lipids and carbohydrates, and besides some proteins. And I think that by the help of the nanomedicine, currently we can, and recently we can surmount and overcome some prolonged disease, such as different kinds of cancers, and even in the tissue engineering field, such as regenerative medicine and cell therapy, we can use these nanoparticles. And also in drug administration, drug delivery systems, we can also use these nanoparticle size. But the challenging and the bottleneck is that.
But sorry, I'm going to interrupt you, but this is not the question I'm going to ask you. The story is that I'm just talking about the component, protein corona component.
Yeah.
The majority. You think that the majority of the protein corona is related to the proteinous layer.
The majority of the things that absorb to the surface of the nanoparticles or even exosomes are proteins, and beside them there are some lipids and some carbohydrates.
Thank you. Thanks for your comments. Great. Mr. Mohammad Nejat, and how do you think about the protein corona?
About protein corona and its role in extracellular vesicle uptake, I can mention that, as we know, extracellular vesicles called exosomes can transport signaling molecules and synthetic cargoes. However, the communication, mediated communication of exosomes can be limited by a coating called protein corona.
Are you exactly sure about it? Do you have any documents about, this statement or not?
The This was a paragraph that I studied in a research recently published in PubMed. So, I mean, I can say that it's valid.
Mrs. Bazdar, can you explain in details how many layers or how much layers, I don't know,
Yes.
is this in protein
As
kernel?
my friend mentioned, that the corona protein is a layer around the nanoparticle sized in the body, such as exosome. And the studies show that there are two layers around the exosomes. In general, the inner hard layer and outer soft layer, which they have interaction with each other. And I want to mention that the protein-protein interaction is in the soft layer of corona.
That's all.
Yes.
Thank you, Alireza. How many layers exist around each nanoparticles? I mean, can you enumerate the layers exist in protein corona?
Actually, as my friend said, there are two layers around nanoparticles. And I have to mention that not any nanoparticles can have protein corona on its surface. There's a size limitation. The nanoparticles above 400 nanometers, mostly they can't have protein corona around it. So there are two layers of protein corona around the nanoparticles, the soft outer layer and the hard inner layer. And the difference is that the hard inner layer is more covalent, has more covalent bondings.
It's stable, you mean?
Yes, it's more stable.
Yeah.
And the soft outer layer is low affinity layer, actually. Their molecules have low affinity.
Can be replaced in any circumstance, in any image and
Yeah, actually
environment.
this soft outer layer in the body fluids, they can change rapidly.
Great.
Yeah, they can change rapidly. And mostly they are used to mention the stealth properties of the nanoparticles.
Great, thank you. Mrs. Narimani, do you think that the formation of protein corona can just affect the bioactivity of each nanosized particles? I mean, I'm just focused on about exosome or extracellular vesicle. I mean that any interaction, reciprocal interaction between their external tetraspanning ligands with their receptor on the target cells.
Thanks, Professor.
You're welcome.
Actually, they can, often in ways that they diminish or alter the interaction between receptor and ligands.
Okay, thank you. Actually, we could not deny the potential of protein corona formation around any nanosized particles. Yeah, absolutely, it can just affect. But we are not certain, or we are not sure it will be beneficial or detrimental in the bioactivity of exosome. But it's a natural phenomenon occurred inside the body. So we should just elaborate different aspects of protein corona on the possible interaction between the tetraspanning. I mean that the surface tetraspanning are incorporated to the exosomal membrane and the possible interaction affinity with their cognate receptor on the target cells. Thanks a lot. Mrs. Salimi, how do you think about protein corona? What's your idea about the protein corona?
A protein corona is a layer of protein that forms on the surface of nanoparticle when they come into contact with the biological fluid and spontaneously adsorb to the surface of nanoparticle, including exosomes. Upon exposure to biological fluid like blood and serum, this complex dynamic coating can alter the physicochemical properties of exosome, including its size, surface charge, and overall biological identity. The formation of protein corona can key processes involved in distribution of exosomes, including their stability, circulation size, and time, and cellular uptake, and targeting specificity.
Thank you. Hesam, I'm going to ask you about the possible effect of protein corona on the size, on the alteration of the size of the exosome or EBS. How do you think about it?
From my point of view, very scientifically, actually, it can have a very specific effect here because the integrity of the protein that caused to with combination of nanoparticles that make the protein corona with the EV content, they can actually do that. And,
enlarging the size
in the
of
large scale and small scale, and also their functions.
Okay, we are believing, in fact, that the formation of the protein corona is going to increase the mean diameter of the exosome. So, I'm going to make a dynamic discussion right now. Any guys, any participants that wants to can be just involved without any limitation. Do you think that the formation of protein corona can, as Hesam told, can just even that affect the internalization mechanism? Because from one aspect, we are believing in fact that protein corona can just increase the mean diameter of exosome and EBS. So by increasing the size of the nano-sized particles, maybe the mechanism which are involved in the internalization of the particles can be altered or not. Any guys? Welcome.
Actually, then I can continue. Actually, it's based on the smart or engineered drug delivery system in the content of in vitro or blood circulation, for example, injection. Also, we can in the clinical condition we can try it, and because of that, the size is very important. So, for example, imagine that we are going to target the specific area that we are going to send the coronary protein to function there. Okay.
Listen, I'm just talking about the exact mechanisms which are activated after making some physical attachment between the exosome and the target cells.
Their surface actually.
And so you are believing in fact that, let's make it clear, you are believing in fact that the protein corona formation is going to just alter any, I don't know, ligand or receptor interaction.
For example, ACE2 receptor, for example, we can it can affect our sleep actual circulation also and rhythm. It's very important, because it's based on fact and studies.
May I raise a question?
Yeah.
I think that entering nanoparticle size to our blood plasma can also alter the interaction between cellular antigens, and sometimes it can be uptaken by the healthy cells. Because of that, I think that sometimes protein corona is not a good idea, and we can need something to stop these situations happening in our body.
But it's a natural phenomenon. We could not just deny it. It's the only thing that a story is going to goes up, and by time we could not stop it.
So sometimes targeting therapy is going to be down, and we cannot targetedly use these our drugs, and we can need some further investigations in this regard. And sometimes also it depends on from person to person. It is going to be vary, and this is another questions in my mind.
Okay, I'm going to just make it clear at the second round. Yeah, absolutely, why not? But my main question is that the protein corona could affect the internalization mechanism. We just hypothesize that we were just speaking about just single cells. Do you think that the formation of protein corona can just influence the internalization mechanism or not? I mean that in general we have several mechanisms which are involved in the internalization of the exosome. For example, ligand and receptor interaction, lipid rafts, micropinocytosis, phagocytosis, even direct fusion. So I'm going to highlight the possible effects of protein corona on their internalization mechanism.
So is it good or bad?
We are going to debate it, I don't know. I'm neutral.
Something?
Yes, absolutely, Eliza, why not? Okay.
First of all, the protein corona, of course, affect the intracellularization, okay? Because it can both affect the physical and chemical aspects of the nanoparticles. Zeta potential, size, diameter, even the chemical compounds around the nanoparticles. So, of course, it can affect. But there is no good or bad effect, okay? You talked about downregulation of target therapy. there are many cases that actually we are trying to use normal cells, not only target the, for example, tumors or cancers. So if it's a natural phenomenon, so we can't say it's a good or bad. Actually, if, as you said, that's a fact, that there is a difference between person to person, from genetical aspects, from biological aspects, body fluids, and many other sections, okay?
Even the materials that we can use, for example,
Yes,
polymers
exactly.
in nanosized, it's going to be different.
That's a potential, of course. That's a potential that can be used in a precise personal principles.
Yes.
Can I add
Yeah,
something?
one by one, every participant. One by one. Yeah, I will return to you after.
Can I add something?
Yeah.
I think we are missing a big point here, and actually this is the immune system in our body.
Yeah, this is the point.
Yeah, I wanted to mention that. Yeah,
But I'm just
Exactly.
saying, yeah,
Actually,
you're
yeah,
exactly
I want to
right.
talk about-
You're exactly right. But I'm going to just highlight this phenomenon. Not are we going to jump to the effect, potential effect of immune cell at our second rounds or third rounds. I'm going to say that even the protein corona can just alter the mechanism of entry into the one single cells. I'm just talking about the one single cells.
Why not? It's carrying. It's like a bioshuttle.
Oh, and so-
of course it will change it.
Okay.
It's obvious. It's crystal clear that the component of the exosome or any nanoparticle which is covered by and shaped by protein corona is carrying something important. Maybe it's carrying a new design drug. Maybe it's carrying a DNA particle or an RNA particle. So yeah, actually it's the target. It goes into the cell. Of course, it will absorb, or maybe the surface and markers or ligands wouldn't let it to enter the cell. Actually, it is I think the main problem in absorbing exosomes.
Okay,
Yeah,
great.
if it goes in, yeah, it works.
You mean sorry, let make a little clear this point that, for example, you're going to mention this fact that a protein corona formation can, for example, change. It's my logic. I don't know. Maybe I am right or I'm not right. So. Protein corona can just alter the mechanism of internalization. For example, it can just switch it from the ligand or receptor to, for example, phagocytosis, even microcyte.
Yeah, of course.
You are right.
Yeah, I'm in the same ground with you about this
Thank
point.
you. Thank
I
you.
have two questions, Professor.
No, just I am going to just see the comments.
Along with the topic that you are asking questions, can we predict these mechanisms beforehand in order to facilitate entering these nanodrugs to the one single cell? And also,
I got it. I got your
and
message.
also, my perspective is from engineering view. I'm not very familiar with the biological view of the topic, and I would like to study more, and in order to learn. And I would like to ask another question. The question is that, can you describe how the interactions happens while entering these
to the best of my knowledge, as I know good about it, I don't know, maybe I'm not absolutely, I know in a recently discovered phenomenon. The different studies, they're going to just isolate and separate the protein components, and then they're going to just identify each component. For example, protein 1, protein 2, protein 3. And then according to the composition, the final composition, they're going to predict how this protein can just interact with the different kinds of cells, Even that any alteration in the protein kernel fingerprint can just predetermine how this particle can just interact with a specific or certain cell lineages. You know what I mean? But yeah, it's a long way to just discover how these particles just can interact with our desired cell or our target cell lineages. Yeah, great. Mrs. Khosravi, any comment?
In the case of targeted drug delivery, of course, this layer can change the targeted efficiency of the exosome.
It's a good or bad?
It's it's bad, of course.
You're certain
Not
about it.
certainly, I can't.
But I
say
could not
so.
stand with this statement, you know.
Yes, but in many cases it can be bad. For example, when design exosomes for targeted drug delivery, and these kinds of protein shapes the outer layer of
great.
exosomes, different kinds of proteins and ligands are on the surface of exosomes.
Great.
Yes, and we designed this exosome for targeted drug delivery. This protein layer cover all ligands, and by this way, it reduces the efficiency of a targeted drug.
A more specific question: do you think that this affects non-target delivery is related exactly closer to the protein corona or other biological activities of exosome? For example, massive biodistribution. We have any control on massive biodistribution of exosome. Exosome can be trapped in, for example, pulmonary systems or other certain, for example, reticulo endothelial apparatus like a spleen and liver. Ali Reza, what do you think? Any idea?
May I say something, Doctor?
Yeah, why not?
I mean, according to what my friend mentioned, that in her idea, it's overall a bad thing. I mean, up to now, there are two strategies that have been introduced, including pre-incubation of exosome with plasma, and then surface modifications in vivo by distribution. Yeah, with these alterations, with this changing engineering, we can overcome this obstacle. So overall, obstacles. Yes.
Yeah, great. Thank you, Mrs. Narimani. Any comments?
I want to say, mention about the interaction between receptor and ligand pathways, different pathways.
Great.
The protein in protein corona may compete with ligands for binding site and receptors.
Great. Can you make any example or not? Okay, go ahead, go on.
And if they have higher affinity than ligand, so they inhibit the ligand interaction and to the receptor.
Some competition exists between some protein corona component with their natural, for example, ligands like tetrahyspanin with their cognate receptor. Great.